Okay, good morning, everyone. So this is the $60 million question. How big is the problem? Well, that in part depends, I think, on where you're living. So, and what we've seen over the past few years is that this is an evolving space. But it's something that I think at WHO and with a number of our partners, we've been taking very seriously. So you saw already some references to different publications. This is a report with Chin Chang and some other colleagues that we published in 2014, when at that time the only thing on the map was these high prevalence of Pfhrp2 and Pfrp3 deletions in Peru, a case report in Brazil, a small study in India, and some evidence of deletions at low prevalence from some archive samples in Mali, and small data set from samples with low prevalence of Pfrp2 deletion in Senegal. So that's where we were in 2014 when we reviewed the literature. And, but we felt at this time that not understanding really what was potentially causing this, and because the stakes of overcalling this or undercalling this are high, we published this paper where we put out some guidance in terms of how to identify, how to investigate, and how to accurately report suspected Pfrp2 deletions. And things were fairly quiet, I'd say, for almost a couple of years. And then in 2016, as has already been pointed out by the last speaker, we started to see reports of Pfrp2 and Pfrp3 deletions, although still at fairly low prevalence, but in Africa. And this, as you all know, is a region that is highly dependent, if not almost exclusively dependent, on Pfrp2-based diagnosis. And so, which wasn't the case in South America. And so, although we felt that this was an emerging problem, we thought this is something that we need to try to get ahead of because it's something that's very difficult to respond to at a patient and provider interface, because the majority of malaria RDTs that are done are negative. And how can you distinguish one that's negative because the patient doesn't have malaria from one that's negative because of an Pfrp2 deletion? So the reports kept coming in 2017, so more recently a case report from Bangladesh, another report of high prevalence of deletions in Eritrea, also now from Rwanda, and then fairly startling results presented in a poster yesterday from Ethiopia. And so I think these reports, which are becoming, if not pretty much almost a monthly or at least a quarterly occurrence, last year at our Malaria Policy Advisory Committee meeting, we had some support that we put together a global action plan. And so first and foremost, in terms of trying to develop an action plan, you do need to know how big the problem is. And so the first order of the day with this global action plan is to define the frequency and distribution of diagnostically relevant Pfrp2 or 3 mutations in circulating falciparum strains. And we chose the words carefully here. We said diagnostically relevant. And so what that means is we're most interested in Pfrp2 and Pfrp3 deletions that are leading to false negative RDTs. And that is not something that we can easily extract from a lot of the papers that have been done, because in some cases we don't have RDT results. So we don't know if the Pfrp2 deletions were actually causing RDTs to be negative, and for clinical cases to actually be missed. So we also, through this global response plan, want to provide concrete guidance to countries on malaria diagnosis and treatment in settings where such mutations have been found to be frequent. We want to identify and articulate the gaps in our knowledge. I think some of those have already been articulated by the last speaker about the genesis and spread of the strains. And we want to identify actions required to develop new accurate tests for malaria-based diagnosis that are based on other target antigens. And so the initial talk was a very nice segue into this. And then ultimately we need to make sure that the communication and the advocacy around this issue is coordinated across all of the key stakeholders. So this global action plan is currently in a draft form. It was reviewed by MPAC just a couple of weeks ago. And really the core components of this are surveillance, laboratory strengthening, research, R&D, and then coordination. So with the number of reports coming out with some frequency, I decided that I would capitalize upon an effort that was already ongoing in our department to develop what we've referred to as malaria threat maps. So these were, it's an online web-based mapping tool that was originally being developed to support data from vector insecticide resistance and for drug resistance, but we've adapted it to incorporate the Pfhrp2, the distribution of Pfhrp2 deletions around the world. And so we developed a database based on reported published reports that led us to find about 130 surveys. So that isn't 130 published reports, but it's basically 130 distinct areas from which samples have been collected and screened for Pfrp2 deletions. And those surveys come from 26 countries. So the map itself, which I'll show you in a second, basically covers the period 1998 to 2017. It includes only published reports of confirmed deletions. So there were some reports we excluded because they were suspected deletions. So we're left with 124 surveys in 24 countries. But already this needs to be updated to include data just published today from Kenya, reporting deletions, as well as I mentioned, one of the poster presentations with data from Ethiopia. So you can then filter the results and change the map based on whether you're looking for just Pfrp2 deletions or the presence of dual deletions, by the clinical status of the patients that were screened, and then also by the survey type. But there's kind of a warning that goes along with interpreting the information presented in these maps, and it's that across the surveys, the criteria for selecting samples to test for deletions varies. So much of the data that's presented on this map is not necessarily the true prevalence. It depends. There's selection bias in some cases in terms of how those samples were collected, and also because it wasn't possible really to find one. In some cases, they don't mention the total number of patients from which these samples were derived. So the kind of common denominator that we could come up to with these maps was to simply look at the number of samples that were tested and then the percentage of those that had Pfrp2 deletions. So for more information, you really need to go to the original resource. So this is what the map looks like. So you can see along, well, maybe you can't see, but along the bottom of the map is the timeline, so spanning from 1998 to 2017. Each place that you see a dot here on the map is where some kind of survey has been conducted, and the size of the dot is related to the number of surveys that have been conducted. So you can see in DRC, for example, where a large survey was done using data from DHS, the dot is quite large. And then, why is it not working? Okay. Okay, so then if you scroll over one of those blue dots, you can then click on that, and then you can get the survey-specific data. So the red means that Pfrp2 deletions have been confirmed in those settings, and gray is where they've not been identified. And then you can go one step farther and click on one of those dots, and then there's a pop-up that describes the type of survey that it was, the year that the data was collected, and then the number of samples that were tested and the percentage of Pfrp2 deletions, Pfrp3 deletions, and dual deletions found in those samples. So we hope to keep this regularly updated. As I said, it needs to be updated already. And the link to these maps is at the bottom of the page. And they are due to essentially be live for all threats, drug resistance, insecticide resistance, and Pfrp2 deletions today. So have a look. There's going to be contact information, so if you see issues or problems with some of this, you can get in touch with us. And we encourage people to share reports with us so that we can then upload that data to the maps and keep them up to date. So what can we conclude so far? So we can conclude that there are clear local hotspots in the Amazonian regions of Peru and Colombia and Eritrea. And then, as I said, based on the poster that I saw yesterday, I think we can also be quite confident that high levels of deletions exist also in Ethiopia. There are surveys that are in the late stages of planning in Ethiopia, in the Hamara and Tigray regions, as well as in Sudan and 2 states bordering Eritrea. And also, I think Jonathan will give an update on a survey being planned in DRC, and then also in Ghana, they're planning surveys. The prevalence, though, of Pfhrp2 deletions varies by location in any given country, and you could probably see by just the screenshots that I showed that there's a mixture of red and gray in these places. And so not only does it vary in its presence and absence, but it also varies in the prevalence. So in many of the studies, however, the methods by which patients were selected resulted in overestimates of the true prevalence of deletion mutants. So if you're reporting prevalence of deletions based on the total discordant samples instead of all microscopy positive cases. So that's why I said so you need to work back to the total number of PF cases that were evaluated and calculate the prevalence based on that, not on the number of discordant samples, meaning Pfrp2 negative, microscopy positive ones. So again, this is just something that you need to be careful when you're looking at the literature. Then depending on the survey design, it's also possible, I think, to underestimate the prevalence of deletions because of the cross-reactivity of Pfrp2 RDTs with Pfrp3, and then also obviously the circulation of strains with deletions which may be masked by co-infection with strains without such deletions. Also an important note is that the absence of PCR amplification may be due to inadequate quantity of parasite DNA, and I think this was one of the issues that we had with some of the original reports, and that many of the studies, the methods used to obtain the DNA did not provide enough material to detect other single copy genes like Pfrp2. So we have produced some guidance around this topic. We published an information note back in May, I think 2016, and then updated that in September this year, where we sort of, this note is more aiming at programs so that they know how to approach this problem. And so one of the important things we wanted to highlight is that there are many different causes of false negative RDTs. So it can have to do with the product design or quality, operator errors, low parasite density of the host, and also transport and storage issues that can compromise the integrity of the RDTs under hot and humid conditions. And then also on that list, but again, in addition to all those other potential causes for false negative results, are the parasite factors like Pfrp2 deletions. And so it's important that with the increasing knowledge and awareness of this issue, that we not always jump to the conclusion that false negative RDTs are attributable to Pfrp2 deletions. And so in this document, we also provide some guidance on how to conduct an initial investigation, both at the patient and at the program level. And so when we say at the patient level, it means how, when I see so many negative RDT results, how can I distinguish one that's due to Pfrp2 deletions from one that's just simply because the patient doesn't have malaria? And this is a challenge at the patient-provider interface, and we don't have an easy solution. But what we suggested is that before you jump to any conclusions, that you get some initial evidence. So in terms of repeating the RDT, sending the patient for quality microscopy so that you can be confident that the RDT is negative but microscopy is positive, or repeating the testing with an alternative RDT that has a different antigen target. And it's really only on the basis of having that kind of initial evidence that you should pursue further investigation in terms of PCR confirmation of Pfhrp2 infection and then subsequent evaluation for amplification of Pfrp2/3 genes. And then at a programmatic level, where we think programs may need to respond to this, is when they are receiving multiple complaints or anecdotal evidence of Pfrp2 deletion. So false negative RDT results being confirmed by microscopy somewhere. And this is actually what happened in Eritrea, where they were getting multiple reports of negative RDTs, and then patients were referred to hospitals where they had microscopy and were found to have high parasite densities. But it is difficult at both the patient-provider interface and on a day-to-day basis to figure out how to distinguish true negative results from false negative ones. And so also in this information note, we provide some suggestion around where to conduct surveys and around alternative RDT options. So essentially what we recommend is that anywhere where there are reports of Pfhrp2 deletions, that these should be followed by surveys in the affected countries and neighboring countries to establish the true prevalence, and that currently alternative RDT options are limited, but I'll come to that in a moment. Okay, so I think, as I mentioned, because of the provider-patient interface, this is a very difficult problem to assess. And because we want to avoid finding reports in the future of very high prevalence of Pfrp2 deletions, we really want to get ahead of this. And what we're suggesting is really a harmonized approach through standard surveys. And so we have developed, and again, it will be released very soon, it was just reviewed at the most recent MPAC, is WHO is providing a kind of standard survey template which is based on identifying these diagnostically relevant Pfrp2 deletions. And so I'm sorry, you probably can't read everything that's here on the screen, but the bottom line is that what we're recommending is that patients are screened for discordance in their between two RDTs, one targeting Pfrp2 and one targeting an alternative antigen like PLDH or PF-specific PLDH or microscopy. And where you find discordance between those two, Pfrp2 negative and either microscopy or PF-PLDH positive, you then consent that patient for collection of blood on filter paper, and only those samples go for PCR confirmation for Pfhrp2 and for to look for Pfrp2 deletions. So we're focusing on symptomatic patients who are presenting to health facilities. We're suggesting that the sampling domain be at the province or state level, and that 370 patients in each domain be enrolled, or sorry, PF patients be enrolled, and that's where you do the dual RDTs, and then you collect blood spots and filter paper from those patients with discordant results. Then you will then end up with three different potential scenarios, and these are outlined here. So once you've enrolled these patients, you have your discordance, you've analyzed those results, you will then calculate the prevalence of Pfrp2 deletions that are causing false negative RDTs. And then the three scenarios basically mean that you will end up with either a confidence interval that overlaps with 5 or includes 5. So if you're below 5%, then we say you need to continue monitoring and repeat the survey in 2 years. If, however, the confidence interval doesn't include 5 because it's above 5, then depending on your this would trigger a change in the RDT nationwide, and this would be prioritized based on the prevalence in other provinces. And so if you find it in one domain or one province, we say that is enough to then trigger a change in RDT across the country. And that is because we believe, and there's some evidence to suggest, that this problem is not one that goes away, but rather that it will spread. And then if you find that your confidence interval includes 5%, then the statistical analysis is inconclusive as to whether or not the true prevalence of Pfrp2 deletion causing false negative results is greater than 5%. And in this case, your options are to continue enrolling patients, to keep a larger sample size, or to repeat the survey in 1 year or 2 years, depending on your resources. Now, that is a survey that the protocol, as I said, will be released shortly, and it is something that WHO will be able to provide technical assistance with, but it will actually be the countries themselves that will be carrying out these surveys. Now, in terms of alternatives for RDTs, the options are currently very limited. In the round 8 of product testing, which is underway, 10 of the 35 products have PF-PLDH targets. And we're planning to release those specific results early in March or April of next year. So currently there are only 2 pan-only tests and 1 combo, that's PF-PLDH and pan-PLDH, that meet performance criteria, and only 1 pan-PLDH test which is actually pre-qualified. And the link there is to the information note that gives an overview of all of the different potential options. Some of these tests need to be assessed against Pfrp2 deleted strains to determine their performance because they include both Pfrp2 and PFP-LDH on the same test line. And fortunately, in this round of round 8, we will be able to do that because we will have those kinds of samples included in the panel. We have, and I've described this as loosened the performance criteria to select RDTs for surveys, because otherwise there would essentially be almost no options that meet our current criteria. But these tests, although they don't meet our current procurement criteria at the high parasite density, 2,000 parasites per microliter, they do. So they do perform well at the higher parasite density, and that's why we think, although we're not recommending them for case management, for the purposes of surveys to identify discordant results in patients, we think that these are adequate. So I'm running out of time here, so I'm going to go quickly through the last few slides, just highlighting some of these additional core components of the global plan. So we think a key component to this plan needs to be laboratory strengthening. So meeting the requirements for capacity to assess the false negative RDTs in countries and during surveys will require quality assured microscopy and staff trained to rapidly read and use correctly non-HRP2 based RDTs that are not currently in use. Obviously, confirming the presence of these genetic deletions requires various procedures from sampling, labeling, preparation of the dried blood spots, and shipping, and then obviously the multiple PCR analyses at regional or international laboratories. This slide, in the Global Action Plan, we map out an estimate for how many molecular assessments we may actually be looking at. So based on the WHO survey protocol, which focuses on discordant results, we've looked at any country where they've reported deletions and their neighboring countries, and then the number of administrative divisions they have, and then estimated based on a 2%, 5%, and 10% discordance rate to come up with an estimated number of molecular assessments that will be required. So this is just one example for Mali with the neighboring countries, the number of administrative districts, and you can see here that means approximately, if there's 10% discordance, around 5,000 molecular assessments that will be required. And globally, again, based on our current reports of Pfrp2 deletion in countries, that globally works out to around 80,000 molecular assessments. So there are a number of international labs that are currently collaborating with us. But we think that countries that are embarking on these surveys, they need to have a plan for molecular assessments that includes the capacity to ship samples internationally. Research, I think, is already, Dr. Wellams has referred to, is going to be a critical component to addressing this problem. We need to understand better the factors that are driving evolution and the spread of these mutations, and we need operational and technical research to simplify the process of identifying them, because any of the laboratory scientists that have been involved in these evaluations can tell you this is a very labor and complex process, and it's something that we need to try to simplify. R&D for new protein targets. I don't think I need to say more. The first speaker's already covered that and already given us an idea about what the potential options are. Also, I'm going to skip over these. So we've identified several priority areas of work. And also, I think at the most recent MPAC meeting, what was identified as the #1 priority area of work was identifying new targets and essentially developing new tests. So the bottom line at this stage is that the full extent of the threat is not yet known. We do know that our alternatives for RDT options are limited. Otherwise, we could just switch to another RDT. That information on prevalence in the world is spotty, and that we must always balance the risk of missed case of falciparum malaria due to deletions against the equal risk of missing cases by changing to a less sensitive RDT, and obviously the longer-term risk of eroding confidence in antigen-based confirmatory testing for malaria. Thank you.
Thank you.